Dr. Naresh Jandrapant. He is from the National Center for Polar Ocean Research, Goa. So please, Dr. Naresh, the floor is yours.
Thank you, George. A step would have been better, actually. And thanks to the committee for inviting me for this talk. And as for many years, I keep on talking about sub-ice geology. And this talk is also centered around that, but with the specific purpose in introducing you to a program which is running through Indian Science Antarctic expeditions, and then thereafter discuss some of the initial results which we have come up with. So I will be talking in 4 contexts, and each of these, please interrupt me in between if you feel that they are not justified. The 4 contexts are as why a sub-ice geology program. A part of it I discussed in the geothermal heat flow presentation in the role of Miss Dev Samriddhi. And then after why, how it is or it can be done, so that probably it cannot be done fully, but in a way that the present level of errors is reduced to an acceptable level, and we have a relatively more reliable output. And then I will say a few things about why the Princess Elizabeth Land or the Emery Ice Shelf area chosen for this particular program, and then I discuss some initial results. So when we talk about why, well, the sub-ice geology is important in the context of the way we look at the stability of especially East Antarctic ice sheet. I talk mainly about East Antarctic ice sheet. Now, it is relevant because when you look at the biggest trough system with definable faults in the solar system, which is little distance away, then compared to Antarctica, which is almost next door from this place, you see that the situation is such that it allows you to build some of the basic geological data, which is extremely relevant in context of East Antarctic ice sheet. So when you can do this, this is a 4,000 kilometer long trough system, a 2,000 kilometer long fault, strike slip fault, etc. You can work out throws, you can work out the soft sediment deformation where there is no, of course, water there, but you can show that. Andean did this in 2012 in his paper, and it always strikes me, I always show this. But when you look at Antarctica, well, you don't see anything of that, especially in the context of the sub-ice geology. It is, again, the EAIS or East Antarctic ice sheet and its relevance in terms of the sea level rise, contribution to sea level rise is very evident. In Kenny's, part is from Kenny, that the contribution of the sea level rise is more than 50% or something like that. But that is not important. What is important, I mean, that is important, but what is important is the errors in these estimations are of very high order, 79–80%, and if you take different climate scenario, as much as 100%. So which means what? Is that what we are estimating is actually, well, not good or not good enough for any kind of intervention or implementation. And it affects a large domain, or it is expected to affect a large spectrum of existing human population. Actually, it's not related to this, and because most of the geologists are sitting here, it's very, very significant also to understand that just around 10,000 years ago, we were hardly 5 million on this planet. And in 10,000 years, we have multiplied to 8 billion plus. So what has happened is that this impact has become even more pronounced. Well, that is a different story, so sometimes else. Well, so when we look at this, and some of these pictures have already been produced in terms of the water bodies which are beneath. Most of the time, the most striking things are the global climate change, warming, and surface melting. But for long, there has been subsurface melting. In fact, it will be a very interesting project later on. Again, I digress because of not being a very focused person, that when we date the water present in these subglacial lakes, how old that is, which will tell us that the subglacial melting has been much before the current aspect of climate global warming as well as anthropogenic influence. So the subglacial melt has many kinds of significance otherwise. But what is important is this. What is important is this part, that this is what is driving most probably the subglacial melting. And this is not uniform. This has a geology or structure has a very strong role to play. When we look at this area where the ice sheet has gone into sea, we know only one control, that the ocean water is going to impact it. So we have a very good cause and effect relationship which is possible to build. But when we look at inside, where there is also melting taking place, we have very little information. So therefore, the significance of sub-ice geology, or if it was a uniform situation beneath the ice, it would have been very easy. But any indicator, indirect indicator which we look at, for example, this BAS map in terms of the gravity, we see that there are very, very significant variations in physical properties which imply that there are variations in the way the material is, that is the composition, also in the way it is placed, that is somewhere there is, we will see some of the, one or two of the data, that there can be lithospheric thinning in some place. What will happen? That will bring the mantle heat up and it will create a higher geothermal heat flux. So these heterogeneities are a significant part of the large terrain which is there beneath the ice. Now, when we look at this, the kind of information which is available to us beneath the ice is indirect. So one can have a gravity, magnetic, or some other kind of input. But when we look at this data, it doesn't also give you or give us that the heterogeneities can be correlated from one to the other. There are differences between what we are interpreting from one kind of indirect data to the other kind of indirect. So there are gaps or variations or differences between different kind of geophysical data. However, I would also like you to remember this GSM. I will discuss this a little bit later. So, the magnetic data discussed yesterday gives us an interpretation which allows us to build the morphology. And the morphology is not simple. If you look at the scale, it's a few thousand meters to sub-I, so it's a very significant scale. And this is on a continent scale. And actually, the implication for what we are talking about is on a scale which is regional, which is actually in terms of the geological units and geological structure. So it is in terms of few kilometers of outcrop or few tens of kilometers or of structures. So it helps, but it doesn't serve the purpose. There are, of course, 2-3% areas where the outcrops are present and one can directly observe. And Simon actually collected all the data. There were two issues. One, that it is restricted to very small areas. And the second, that it has, all of this data itself has limitation, or that is, there are different degrees of reliability to this data. Well, so that is a problem. So what do we do actually? One of the ways that we can look at the adjacent past terrains and project some of the things which are present, some of the structures which are present, some of the mountain belts which are present, and interpret. So for that, of course, one can look at, and there were some discussions yesterday, quite far back, but I am saying that let us look at the Rodinia, Gondwana, that sort of time frame where there is a reasonable level of reliability. So when we look at that, and one of the, it's just for the general purpose, so one of the compilation when we look at 2005 one, we see that there is a continuity of structures which can give us information about what is present beneath the Antarctic ice. However, when you look at this, you see that the adjacent land information is limited to the coastal or inner domains only, or not to the inner domains, only to the outer domains. Now, this leaves a very large area. Remember, it's really a continent scale area, a very large area without any source. So when we look at this, and there were some very gross correlations made by Boger, 2011 did that. So when we look at, magnify this on a slightly larger scale, in our context of the East Antarctica, we have three major adjacent terrains, that is the Australian, the Indian, and the African entity terrains. In some of these areas, in some of these terrains, there are some work which has been done, and it gives us a good, reasonably good confidence. However, look at the correlation Australian affinity terrain. Well, it is a swath across the continent. It has very, very little or limited variability. With that also, there are a significant central part of the continent about which we have practically, even in this gross correlation, it says that there are rocks with no known affinities. Well, when we come, I just show for a minute the area where we will be discussing something more little later. So this is that area we are talking about, but the time is little less or will be less, so I skip that and come back to this later. One of the terrains which Joachim has worked for long has relatively, is in the droning modeling sector, relatively better control worked out through various means, though not all means were employed, and there is still scope of work. So that is one terrain I just show to say or to illustrate that these areas, that there is a possibility of doing things. So, if this is a problem, and it looks really difficult, how do we actually go about reducing the error or increasing the reliability? So I classify 3 methods. One is direct, which is that of the direct observation of the outcrops. The other is indirect, which is not knowing or not getting any actual material which is present, geological material which is present beneath the ice, and having either remote sensing or geophysical data. So that is indirect. And the third is direct, indirect, which is the displaced material which is there inside, but which can be tracked or linked to that domain, and if it can be sourced outside. So what are those sources? The sources are proximal marine sediments, the moraines, of course in well-defined drainages, and entrained or entrapped material within the glaciers. So we have these 3 avenues which allow us to reduce the errors in understanding subice geology. Well, so we look at, for example, the way these direct exposures can be explored, and I do not spend much time, but I show an area where the example of this work will be shown. When we look at the indirect methods, we have the remote sensing ones and the geophysical ones. And I classify between the two because in the remote sensing ones are dominantly in terms of the hydrology or subglacial hydrology context. And the geophysical ones are essentially looking at the property of the material which is below. So one example of the remote sensing one is this area of PEL, which was not well understood, well explained, well before the second ice cap, and then there is a much better resolution which one sees after the ice cap flights. And the other is that of the geophysical, where you have a relatively much higher resolution magnetic data which has become available now. So then we come to the direct, indirect methods, that is the sources from where we can have the information about subice geology. These includes the proximal marine sediments, and they can be either at the ground or close to the grounding line, or they could be taken a little bit far away through icebergs. So there could be ice-rafted debris, including coarse material, as well as fine grain. So one can have, if there is a good control of the currents and the source, this can be a very good source. And I'll show you some work based on this later. The control, basically in terms of the ice drainages, is very important. So when we look at this, you see this area in Landing Bluff is devoid of practically depositional material. So there is dominantly the signal which you have is of something which has been eroded away. And that is because if you look at the drainages, they don't end there. So there is a kind of indirect verification of the interpreted drainages, which show that where the drainages are present, you have sediments which are being sourced from inside, and otherwise you have a situation like this. The direct, indirect sediments, there can be an array of these sediments, which includes the moraine samples from the terminal end of the moraines, the end glacial sediments, as well as the marine sediments. And I show you. So these are some of the marine sediments which are there. This represent the work done, as I will be explaining a little later. Then endglacial sediment layers here. The most important in these we found are the rock fragments. If we have rock fragments, they give us the best or the most important information. So we come to the next part of why choosing the domain of MA, the ice shelf. Well, we see that yesterday, also in first rows presentation, we looked at some of the areas here. But it is very important because it is an important part of the basin, Emery-Lambert Basin, and it is 16% of that basin. Not only that, it has the ice movement which ranges from practically non-existent to more than 1,000 meters per year. So there are drivers of that ice, possibly from beneath, which have created such a different or such a strong imbalance or heterogeneity in the ice movement. So the reasons for that will be important to see. Then in the coastal tracts locally, there have been very high heat flow reported. Carson's work in 2014 is one example, but later on also, when there is a hole drilled or rare, at the base, the heat flow measurements are much higher than what has been predicted by most of the modeling. So there are reasons of complexity. What are those reasons? Is that there are well-known rift systems there, and the Lambert Glacier itself is in a rifted basin. And I'll explain by an analog structural experiment that there are other rifts which are associated. Then geophysically also, the East African rift system has been described from this area. So there are both compressional orogenic belts as well as extensional component. Both can lead to significant change in properties which will impact the overlying ice sheet. And most importantly, we had no money to do this project. The project was to be initiated within the limitations of the existing Indian Science Antarctic program. So therefore, that also played a part in choosing PL. The Indian station Bharati is there in that area. So we chose an area of whatever helicopter reach can give, a 400-kilometer radius circle towards landward mainly. Now, in this, the geological reasons, I have explained, but it is also in many ways a least explored area. In many ways, many orogenies have been reported, which means that there have been many mountain belts. The rifts which are present there are reactivated rifts. From Lambert also, there have been several instances of magmatism. So there are the extensional domains are not the ones which once created and remain like that. So there have been a lot of variability. And then when you look at this picture here, this is basically predicted lithosphere-asthenosphere boundary depth model for Antarctica, a relatively recent one. And you see that there is quite a lot of variation and in the area in which we are targeting. So there were good reasons to look at or to focus at this area for making some investigation to make inputs for geological, sub-ice geology, to have a relatively higher reliability geological map. The way the tools were there, the way in terms of objectives included untangling the orogenies. So there was more than one event of mountain building definitely present there. Then look at, because it was also facilitated in terms of, because this is adjacent part of India, a significant part. So therefore, it was much easier to look at in detail, in refined way, whether the extension of the structures or structural fabric exists across the two continents, which were earlier together. And there was a important thing, I said, I'll come back to you. The GSM is present in the hinterland of this area. It's really a very high area. Like what possibly is happening in Greenland, if I can refer to that, is that there are high mountains and the slopes are there. And the high mountain slopes and friction and things combined could lead to what is higher rate of ice mass wasting. So it could also possibly mirror that kind of a situation. So these are the things which we were looking at. Well, so when we began a few years ago, and I have some results to show, I'm not saying that we have been able to do enough, but using this premise which I discussed, we look at some of the results now. So this is looking at the direct method of examining the outcrops which are present in the mainly coastal domain of the area. One area which has been examined very nicely is this and West Fold Hills. And we have some new information from this area. So I will classify this in two domains for the purpose of discussion. This has domain 1, which is closer to the Emery Ice Shear, and the other is West Fold Hills. So in this, there has been a detailed examination of about a 70-kilometer long coastal domain. It provides very interesting result. What it shows us, one, that there is an assemblage. There is an assemblage which represents conditions which are unusual. Unusual in the sense that they describe ultra-high temperature metamorphism. So what it actually tells in a more general term is that at some point of time, the geothermal gradient in Earth was very different from what it normally is in most of the continental areas, to cause a temperature reaching up to 950 degrees centigrade or 900 degrees centigrade. So those conditions are well exemplified by the assemblages which are present in these rocks, which means there is a relatively low pressure mineral cordierite forming around the garnets. Cordierite also present in matrix. Then there are associations like corundum, spinel associations. So all these signify texturally or mineralogically the idea that these rocks experienced over a 70-kilometer domain ultra-high temperature. And when we model these in terms of describing a phase equilibria, they can be modeled. So there is a textural evidence which can be confirmed by conventional thermobarometry. Then there is a modeling way in which one can predict when these assemblages which are present on ground will occur and from what precursor they will occur. When we look at that sort of data, you see that these diagrams represent several sections across that 70-kilometer domain. And when you look at this diagram, you see A to B, A to B, A to B line. Now what is this A to B line is telling us, is very important, is that if you look at the vertical axis, that is the pressure. So it is telling us that there is a drop of drop of 5 kilobar pressure, which means around 15, more than 15 kilometer of uplift, suddenly when there is a change from assemblage A to assemblage C here, assemblage A to this white area here. So across a relatively longer stretch of the coastal area, you have as the conditions describing that there was a drop of pressure, which means what? That there was a sudden uplift of 15 kilometer or more across a reasonable length. So this, the description of the ultra-high temperature metamorphism and the decompression or uplift over a longer distance is one highlight. Now this is reflected by or corroborated by the assemblage also. So you see here that this is the higher pressure and it has reduced from 10, 11 to around 5, 6. Well, when this occurred is also contained in the minerals which are present along with this assemblage. These are not separated minerals. They are constrained by the development of these. And they signify two periods especially in this area. The one represents around 500 million years and the other, which has not been well reported, is around 700 to 800 million years. So now, normally these processes are associated with mountain building or compression. The other significance of the ultra-high temperature is that it will lead to a condition where part of the mineral assemblage will melt, and that will create granite. And the granite becomes a high heat producing source. So if that granite is emplaced at different structural level, it will also control the supply of geothermal heat flux. So these are related in that way. Now, so what the relatively newer information tells us is that we have a situation where there was a mountain building activity which ended with a significant uplift and conditions which were good for production of lot of granite, which could lead to a higher geothermal heat flux in that domain, in that mountain belt domain. Okay. Well, normally when you have a compression, it is also associated with fabric development. So when we look at fabric development, there are two main fabrics which are associated with these ages. One is an east-west trending fabric, and the other is a north-northeast, southwest trending fabric. So you see these in rocks. These are measured in rock. And when you actually look at this, in the Bradsand area, in a magnetic map, it may be a little bit speculative, but when you look at these, you find that the magnetic trends also match these two trends. So what it actually means in bigger terms is that it is telling us that there are two mountain building activities at 500 and 700 to 800 million years. The 500 one is well known from that area, and this is normally classified as extension of Kunga or Kunga orogeny, but the way it propagates has been debated differently. That is, whether it is parallel to the coast or it's across the coast. So here, this work shows that there is a arm, or there is at least one 500 million year Pan-African orogeny which cuts across, and its evidence we should find in India also across. So which has also been done, and I'll discuss that quickly. Now, when we look at, as I said, the microstructures which are similar, which are observed there by very detailed structural work, and their extension also, we correlate with the Eastern Ghat of India. And then these are dated also. There has been a recent geophysical survey. Now, if this was there, if this was a suture, it would also reflect at depth in some geophysical signature. So there was a geophysical survey along what we proposed as the Pan-African suture, and we see that there is a Moho offset almost along at that direction. So this is a corroboration. Then I come to quickly to the domain 2, that is the Westfold Hills, which is a very well-known Archean terrain. But here also we found the conditions through modeling, the conditions which were ripe for generation, melting of charnokites, which takes place at high temperature, which again an ultra-high temperature event, and generation of granites. And that granite we dated, and it comes to be Paleoproterozoic age or a younger age. So this happened during around 22 million years ago, 2200 million years ago. We look at the geophysical data integration and the geology structure. So there is one idea or one thing which was known, that the Archean terrain Westfold here towards east, and as you go, a slightly younger or younging sequence. So there was one domain which is called as Rohr group. Where is the boundary between the Westfold and the Rohr group? So this is the data from ice cap, from where there was some suggestion that this is the boundary. But we looked at the structural data and combined the geophysical data. Here there is a gravity data. You see a change in the direction of the gravity contours beneath Sausdale Glacier. So combining all this data, this new information tells us that it is probably here, the source, the Rohr-Westfold contact. So that is another thing which we did. I have some time. Okay, so we'll quickly discuss the two other aspects, that is the subice hydrology and the marginal sediment information. So in subice hydrology, we did an experiment, analog structural experiment, scaled experiment, to talk about or to work out how these canyons and structures develop. For this, we created a rigid block of landing bluff, a material which is similar in properties, and we used a surface which reflected the anisotropies of the basement. And that allowed us to conclude that there can be a 90-degree shift, orthogonal shift to the Lambert will produce a 45-degree system of shears. Okay, so this has, I will not repeat this because I need to discuss something more, but this has implications for the ice sheet in this terrain, or the fate of the ice sheet from this terrain. Now in this area, we collected the offshore sediments from 3 ODP sites. You can see that 2 sites, 739, 742, come dominantly from the Lambert Glacier flow, and 740 is out of that. So we saw the heavy minerals and the rock fragments. So what we get from the detrital monazites and the rock sediments, that the inference of a Pan-African, that influence is very clearly there. The rock fragments give us, importantly, the rock fragments give us an age which is around 700 also. So therefore, there is the influence of the mountain building activity is probably there, a little bit in the hinterland too. Now this rock fragment, which gave the idea, doesn't matches with the outcrops which are present. That is, it is a relatively low-grade metamorphic rocks. And the sourcing, if we see the way it is coming could be from the foothills of the Gambutso Mountains. We also did the zircons in this, and in zircons we get many ages. I'll explain to you why quickly, but if you look at these blue dots, they represent site 740, and they are more towards collision site, whereas, and this and other geochemistry indicated that confirmed the flow lines or supply lines in that area. So when we look at the zircon data, which is a resistant mineral which carries, we see the presence of various ages, including Archean ages, reflects well with the source which has been predicted for that area. Similarly, in the domain 2, we had low-grade material present there. Okay, so I will just finish in a couple of minutes, yes. So we have, in the process of trying to go deeper inside to work this out better, there are some logistic arrangements which have been made. I will not repeat the summary of some of the things which initial results have suggested and shown, but I will say that the dream of a reliable sub-ice geology is still some time away. And these are some of the people, I mean, I did nothing. So these are some of the people who work there in that area, and I thank you all.
Okay, thank you. We have time for 2, 3 questions. Please, somebody. Okay. Well, I have one, probably totally out of this. But you mentioned at the beginning something about subglacier lakes in the Antarctica. And came to my mind those subglaciers, they are there, but nothing to do with climate change, actually. But I'm wondering if right now there is some indication if those subglaciers are increasing, decreasing, or are they stable?
I do not have the ice mass, but what I try to hint at in discussion as a future this thing is that the glacier, the melting or the effect of the warming may not be very simplistic, the atmosphere and sea surface increase in temperature and melting of East Antarctic ice. It's probably much more complex. One way it can be proved, as I said, is that if we can date the subglacial water, the age of the water. And I mean, I don't do that actually, but there are people who are going to drill, and I believe that it is something which can be done. So we should be able to separate the surface global climate change effect from what is a geological context effect. That is what I meant actually.
Okay, so there is another question around? No? Well, we thanks once again, Professor. Thank you. Well, and now I have the honor to present you Linda Amberth. Yeah. She is from the Institute of Marine and Antarctic Studies from the University of Tasmania, Australia. Please, the floor is yours.
Good morning. I'll be talking about ancient DNA around Antarctica. And this is really a research field I've been specializing in over the last 10 years, which sounds really long to me, but it's still a really young research field. And so for today, I've put together a bit of an overview about where this research field is at, how do we recover ancient DNA from the seafloor around Antarctica, what are the challenges, what are the techniques we use, and a little bit of an overview of the research we do at our lab in Hobart in Tasmania. So let's start with what is ancient DNA actually. Ancient DNA is DNA from dead organisms. So there's lots of organisms living in the sea, and when they die, their remains and their DNA, they sink to the seafloor, and the DNA becomes preserved there over really long time scales. So this is great because when we look at ancient DNA from the seafloor, it's a bit like doing forensics really on the seafloor, and we can look at communities across the entire food web. So we can see signals of viruses, we can see signals of bacteria, we can look at eukaryotes, phytoplankton, zooplankton, fishes, all in one go. And that's great, but there's also a lot of caveats that we have to be aware of when we're analyzing ancient DNA. So because it's DNA from dead organisms, it means that the mechanisms in the cell that keep DNA intact while organisms are alive, they're no longer active. And so the DNA degrades really quickly and it fragments. And so the DNA pieces that we find in the seafloor, there's very few of them to start with. They're also really, really short, and they also really degraded. And that makes it quite difficult to analyze it. So what I'm showing you on that picture there is something you should never ever do when you're sampling for ancient DNA, to go in the sediments with your bare hands. It's a very bad idea. We are dealing with such low amounts of DNA that if you did that and you got a little bit of DNA into the sample, whether that's from your hands or the hair or just from the environment, it would override the signal of the ancient communities really, really quickly in the sequencing data. To put that a little bit into relation, when we're looking at the ancient DNA fragments, they're usually less than about 100 base pairs, which is really short. When we're looking at DNA from intact organisms or living organisms, what we're analyzing there is DNA that has length of a few hundreds of base pairs or thousands of base pairs even. So we have to be quite careful of contamination. We also need to make sure that we're authenticating the DNA. And there's a couple of techniques we can use that I'll show you in a few minutes. Antarctica really is a very good place to study ancient DNA. Well, first of all, it's really interesting because Antarctica is changing. It's important. We want to know how organisms respond to this change that Antarctica and the ocean around it is experiencing. So it's a really important study region for that part, but it's also a really good place to study ancient DNA because of technical reasons. So when we're looking at the seafloor around Antarctica, it's quite cold all the time. There's no UV irradiation at the seafloor. We have very low oxygen concentrations, and all of those are factors that mean that DNA gets preserved really well. And there's a reason why the oldest records of environmental ancient DNA are from polar regions. So we've been working on 1 million-year-old ancient DNA in Antarctica, and the latest record of ancient DNA recovery is DNA that's 2 million years old from the Arctic. There are several questions we can ask with ancient DNA in mind. The first one is, how did marine organisms respond to climate change? And what we're looking at here really is to study community composition and changes in the composition through time. And that really is where the majority of research in environmental ancient DNA is focused at. We're looking at records of past organisms through time, and we're using taxonomic information that's preserved. But I'm also personally really quite interested in the adaptational and the evolutionary aspects of this kind of research. So we have a new project that we have just started with a team of collaborators in Australia, where we are pursuing slightly different questions too, such as looking into genomic changes through time. Can we actually detect mutations and genomic changes in organisms over longer time scales? When do we detect them? Can we detect them in relation to environmental change? And if we do detect them, how quickly do they actually get manifested in the genome as a response to environmental change? So we have put together quite an extensive data set. There's sediment cores that we're using for this research from 11 Antarctic expeditions. Half of them I've participated in myself, and so I've been very busy collecting samples for ancient DNA research all around Antarctica. But I also have been talking with a lot of collaborators who are sending samples for this research. So if you do think that you might have sediments that could be used for this, I'd be really happy to hear from you. There's of course a few things we need to consider when we're collecting these samples. So how do we go about collecting these samples? Well, usually we go on the bigger research ships, so we can access the sediments in the deep ocean. For example, here you can see a picture of the JOIDIS Resolution. I've been very lucky to be on an expedition in 2019 on this ship. This was IODP Expedition 382, Iceberg Alley to the Scotia Sea. And so, depending on the research vessel, we really have different opportunities, or we can have different approaches of collecting samples and making sure that we're addressing this issue of contamination. On the JOIDIS Resolution, we can do piston coring, which is the preferred method of acquiring sediments for ancient DNA research, because you're basically punching a hole into the sediments, and we're taking those out, and they often, if we're lucky, have very nicely preserved sediment layers and soft sediments, which is what we need. The problem with piston coring is that it is hydraulically triggered. So we are basically pumping drill fluid towards the piston core, and drill fluid is a really nice way of saying dirty seawater. And so that's really what we don't want at all. Normally we're not getting any of that near our core, but of course for this type of research we really want to make sure that we don't. And we can't avoid it, we have to use drill fluid, otherwise the system won't work. But what we can do, and what we did on IODP Expedition 382, is to infuse this drill fluid with a chemical tracer, PFMD in this case. And so if we do that, we can then measure PFMD traces along the entire core once it is on board, and we can make an assessment of whether there might potentially be contamination in our samples. This is a technique that's very routinely used for microbiological research on the JOIDIS Resolution, or BOS. And in this case, we've applied this to ancient DNA research for the first time. So then we were collecting piston cores up to a little bit more than 170 meters deep. I've collected samples very opportunistically on this voyage, but every 1.5 meter in these sediments. And then the co-chiefs were also super cooperative, and even made it possible that we collect 2 cores of 10 meters in the surface sediments just for the purpose of ancient DNA sampling, so that we could sample and split and sample them on board, which gave us a high resolution record of the more recent sediments. Yeah, but if you're on a different ship, the situation is completely different. So what I'm showing you here are some pictures of the Australian research ship, the RV Investigator. I've been on voyages to collect samples on this ship multiple times, once in 2017 to the Totten Glacier in East Antarctica, in 2023 to Cape Darnley, also East Antarctica, and we have a voyage coming up in 2026 to go to the Cook and Ninnis region again. So it's a multidisciplinary research vessel, means it's not entirely dedicated to drilling. The sediment cores we take here are much shorter than what the Joides could do. So we're looking at multi-cores, custom cores, piston cores, up to about 24 meters. So you can see our sampling here. We're always very careful, like dressing up with all sorts of things to avoid to get DNA into our samples. I've put here the pictures of a little protocol that we've been developing in case anybody's interested to integrate this into their research and collect samples for ancient DNA. This is a very easy to use guide on how you could do this. It's one of the first products of the SCAR Action Group ICE Pro. We don't only have that for ancient DNA, but also for microfossils, for GDGTs, and working on carbon dating sampling as well. I think if you're interested in this, Johan is here. He's the lead of the group, so please get in touch with him. Yes, so like I said, most of the time I've been on research voyages and sampling in the deep ocean or on the continental shelf, but last field season, 24-25, I was actually able to go to a very different field campaign and participate in the SWACE 2C project. And SWACE 2C stands for Sensitivity of the West Antarctic Ice Sheet to 2 degrees of warming. So this is a very big international project. You can see the project leads I've listed here. We went to the Ross Sea ice shelf and then actually drilled through, well, you know, the drillers did, drilled through 600 meters of ice, almost. And then we were collecting sediment cores underneath. So this is really an environment I've never looked at before, so I was very excited to get into the lab right away and test if we can find any DNA in these kinds of environments. You can see we've been sampling very carefully here. We had a container that we set up to be able to slice the sediment cores that we got. So back in the lab, we did some first extraction tests, and that was really because, yeah, it was such a different environment. We just wanted to see if one method works better than another when we're extracting the DNA. So what you can see is some preliminary data here from just a few samples where we were comparing community composition using 2 different protocols. And so the left protocol, I'm really glad we did that because one of the protocols didn't really work at all. And so we're now going with this protocol. I've just called this method B here for the moment. But yeah, we did find a lot of different signals from, including diatoms, phytoplankton, other organisms, as well as invertebrates. So this looks really promising. And Jacob Bradford is a master student in the group who's now gone and extracted DNA from almost every centimeter samples that we had throughout the course. So this data is being sequenced as we speak. But we can go back, and so we're in the field, we're collecting the samples. What we normally do is freeze them straight away. And then under frozen conditions, we transport them to the lab. So over the last 3-ish years, I've been setting up an ancient DNA lab at IMAS, the institute where I work. And this is what it looks like. So you've got an entry room where we put on PPE. We're getting more and more paranoid along the way. You can see on the second picture there is one of my students in a hazmat suit. So we're really taking all of the precautions we can to protect our samples. And so this inner lab is positively pressured, which means that all the air is transported away from the sample. We have very frequent and stringent cleaning regimes in this lab. When we are extracting DNA, we always include several controls. This is to track cross-contamination or any contamination that could potentially have gotten in the sample. We've got a number of techniques or protocols to extract DNA that we routinely use in the lab. And I just wanted to show you my new little toy that I have, which makes setting up reactions in the lab so much easier. If you do this, every day for hours and hours with pipettes, these little robots are just amazing. Okay, so then how do we go about analyzing ancient DNA? Usually, we go for a technique that is called metagenomics. And so that means that we're basically extracting all the DNA in our sample, and then we are sequencing it randomly. And so because we're sequencing it randomly, that's often why it gets referred to shotgun sequencing. So it's the same thing when you hear about this. There's a couple of advantages when we're using this technique. So first one is that we are keeping the communities in proportion. So we're basically looking at everything as it was in the sample. The other very important advantage of it is that we're keeping DNA fragments as they are and the DNA size distribution in our samples. That's important because that is something we use for authentication of the ancient DNA to show that it's really ancient and it's not contamination by modern organisms. Yeah, there's a little, I will explain that in a little bit more detail. So like I said, metagenomics is this random sequencing of everything in the sample. What you are seeing here on this image on the bottom left is like a size distribution of DNA fragments in an ancient DNA extract. So you can see the fragments, most of the fragments are really, really small, less than 100 base pairs. There's another technique that is very often used in genomics research, and it's fantastic if you're looking at modern organisms and intact DNA. That's called metabarcoding. So here you can actually select certain organisms that you're interested in. They design little probes that attach to the DNA in two places, and then we can basically amplify and copy everything that's in between on this piece of DNA. We amplify it, we sequence it, and we can look at community compositions that way. The problem here is that usually these fragments that you can look at are larger than 100 base pairs, and they're all the same size. So if we use this technique, we're really amplifying this part of the community here, which isn't really a good representation of our sample at all. So it's really important that we're doing this random amplification of DNA in the ancient DNA research area. There's a variation of metagenomics, or let me first say, well, metagenomics also has disadvantages because it's really expensive. So if you are amplifying and sequencing everything in your sample, but you are only interested, say, in diatoms, which might be like 0.01% of the entire community, you pay $10,000 or so for a sequencing run, then that's a really huge waste of money. And so that's not ideal in many cases. So what we can do is a targeted approach, like the metabarcoding approach, but where we're preserving the size variation of the DNA fragments. That's called hybridization capture or target capture. Here in this technique, we're designing RNA probes that we can design so they're complementary to the DNA stretches that we're interested in. And then we have our DNA extract, the RNA probes. Basically, we throw them into the extract, and then we're fishing them out. That's why we often call them RNA baits, because it's like when you're going fishing. So we're taking the DNA out of the sample, amplifying it, and then we're looking at those particular target sequences. And they can be designed to anything that you like. It could be one gene of interest, it could be 5 genes of interest, it could be a whole genome or multiple genomes. So it's really flexible as long as there's a reference sequence for what you want to look at. So in terms of authentication, there's different techniques we can use to really make sure that the DNA is ancient. The first one I've already described a little bit. We're looking at the size distribution of DNA fragments in our extracts. And so here the hypothesis is that the older the sediments are, the shorter the DNA fragments get. But it's not really that simple. There's lots of parameters that influence DNA fragmentation, and if you're interested to read more about this, I really recommend to look at Caitlin Selway's paper from 2022, where she was investigating samples, archive samples from IUDP to address this. A much better way of authenticating ancient DNA is DNA damage analysis. And so what you're doing here is you can imagine you've got your DNA fragment. The ends of the DNA fragments are exposed to the environment, and so they're quite vulnerable. So there's chemical modifications that are happening over time, substitutions in the bases of the DNA. And then this is something we can look at when we have the sequences using computational tools. So we can measure these rates of substitutions. And if the frequency of these substitutions get higher and higher, it means they're more and more damaged, then we get these damage plots that you can see that they have this typical U-shaped curve. And this is a very good way of authenticating ancient DNA. So this is exactly the approach that we used for the IODP samples. We measured the DNA damage with depth at the 3 different sites that we were collecting sediment cores during IODP expedition 382. So you can see there's an increase in DNA damage with depth at all 3 sites that we were investigating. We've done a couple of additional correlation analyses between DNA damage and other geochemical parameters that we had that were collected on board. And what we found is there's a very strong correlation with ammonia, with alkalinity, phosphate, so signals of organic matter decomposition. There's different programs that can be used to do these damage assessments. In our case, we use something called HOPS, but there's map damage and different techniques that can be used. And then, of course, we know that the DNA is ancient. What we really want to know is what are the organisms that we're looking at. And so this is like a typical image of the first kind of results that we get to see when we're looking at the data. And if you think that's a lot of organisms, that's true, but it's even only the eukaryotes. So we're literally seeing hundreds, if not thousands, of species signals in these samples. And then we have to make sense of it. And what I've plotted here is the eukaryote composition at the 3 IODP 382 expedition sites. They called U1534, U1536, U1538, the Falkland Plateau, and the Scotia Sea. And so I've plotted the eukaryote relative abundances with depth. And when we did that, what was really exciting was that you can see here at U1536, we have samples that are dated to about 1 million years. And that was amazing because we had just recovered like 1 million years old ancient DNA from eukaryotes, which was really exciting at the time because up until then, the oldest record of ancient DNA in these kinds of environments was 140,000 years. So we really expanded the timescales, and we can look over now glacial, interglacial cycles. It was exciting while it lasted. I just told you about this 2 million year old record from the Arctic, which was published 2 months later. But yeah, it was great to find this. What you also see here is some clear transitions in communities that we found. So you can see there's a very strong increase in diatoms, which is this olive colored here, after approximately 14,500 years ago. And so we know that's a time where Antarctica underwent significant changes. And we've related this to changes in the water column following meltwater pulse 1A. What you also see on this graph here is that at site U1536, we kind of see these trends, but we have a big gap in data here. And this is really what kind of sparked that we want to look at these records at much higher resolution and at many more sites around Antarctica to see if we can find the similar trends. Even if we're looking at site U1538, there's still like 2,000 years or so in between the samples. And I'm really interested to look into this in much more detail. So we've started on this new project, and we started with the high-resolution record that we've also collected at IODP Expedition 3A2. You can see this is a completely different core here on the left, but the same site, and the results are very comparable. So again, we see a very high increase in diatoms, this turquoise color, around this time, 14,500 years ago, compared to other eukaryotic organisms. At the East Antarctic sites, so these here are from the Totten Glacier, and we had 2 different custom cores. You don't really see that signal so clearly. There's a little peak of diatom abundance at around 8,500 years at both sites, but it's much more short-lived, well, short-lived on a geological scale. What's also interesting in these records when we're comparing them is here you see quite a bit of pink. And that is a signal from Haptista, Theocystis antarctica. It's not an organism that fossilizes, it's a soft-bodied organism, so it's really interesting to find these signals because we can see how one productive organism that is now very productive in the ocean is represented in the ancient, did it just turn off and on? Magic. Is represented here. We don't see so much of this organism at the East Antarctic site. What we do see, though, is quite a bit of fungi. And so that's quite different. So we did look at the damage at these sites also. And first of all, I've been looking at the damage patterns of all of the eukaryotes together. And what you would expect, of course, the deeper the sediments are, the older the sediments are, the more damage there is. But we only really found that at the IODP sites when we're looking at all the eukaryotes. In the East Antarctic, what we're seeing is a decrease in DNA damage with depth, which is really counterintuitive. And so knowing that there's quite a big variation in community composition at the different sites, we started to disentangle that a little and just look at diatoms specifically and their damage patterns. And so this is photosynthetic organisms. If we have these represented in our ancient DNA sample in the seafloor, they should really be ancient organisms. And we do see an increase in damage with depth if we do that, if we separate them out and look at them separately. With fungi, it's different. Fungi consistently usually show decreased damage signals with depth, and that really tells me that they're not necessarily ancient organisms. Fungi also live in the sediments, so it's not too much of a surprise that we're finding living organisms here or, mixed in modern DNA with the ancient DNA, but it is clearly diluting the damage signal when we're looking at all organisms together. I've been talking a lot about like phytoplankton and fungi, but what we also started to look at in the lab is much bigger organisms. So Eliza Davis, she's a PhD student in the lab, and she basically, she's a bioinformatician, and she just took this entire IODP Expedition 382 data set and reanalyzed it. And she put together new databases. She's looking for lots of references for Southern Ocean vertebrates. And so she's been looking at this data set for the last couple of months and was actually able to find some sequences of Southern Ocean vertebrates, including fishes and penguins and so on. This is something that she's actively working on. It's a very small fraction, but it's interesting to see that, even though we have this data generated already, we use different and new bioinformatic tools to look at this data set again, and we can find these signals. So just to summarize a bit what I've been talking about so far. We can track shifts in community composition over time. We can detect DNA of organisms that do not leave fossils behind, small organisms as well as bigger organisms. The record of ancient environmental DNA recovery sits at the moment at about 2 million years. But given that we always thought that DNA doesn't preserve for longer than 1.5 million years, this is very, very interesting, and at the moment the field isn't entirely clear what the actual ancient preservation limit for DNA is. So it's possible we still find older DNA. I think it's very important to say that this also has limitations, this type of analysis, and we need to be aware of the biases. We need to be aware that there's degradation and fragmentation happening, and to make sure that we're interpreting what we're getting back in terms of community composition very realistically. And it's quite a fast-moving field with technologies constantly being developed, but new bioinformatic tools. So it's moving quite fast. And in the last couple of slides, I'd just like to show you some of the avenues we're taking in the lab and projects that we at the moment have. So these are my students. We have a few PhD students and master students in the lab, and you can see the projects that they all have. They kind of all over the place, but they're really targeting different levels of the food web. So working on diatoms, Antarctic krill, mollusks, and different regions around Antarctica. Lucinda here, she's actually just been on IODP expedition 403, and that went to the Arctic rather than the Antarctic. So she's doing something different, and actually feels like she always does. She's always going off the rails a little. We can't control her. And now she is working on ancient RNA. She really wants to know if we can extract ancient RNA from sediments. This is a big challenge because RNA is even less well preserved than DNA. It's very fragile. And so she's done her first few tests. She's already developed a protocol to hopefully extract RNA. Well, it did work. She did get some sequencing results, but she's at the moment working through whether these are truly ancient and whether they are RNA at all in the first place, because we also need to disentangle and see what's RNA, what's DNA potentially. So it's work in progress. In terms of the evolutionary angles, I've been telling you also about this hybridization target capture technique that is very, very suitable. And so this is an approach we're taking for some of the projects also. We're designing these baits specific for certain organisms, say sea ice diatoms or Antarctic krill. And then we're pulling out those sequences of those organisms of the extracts, and then we're amplifying them, and then we're sequencing them, and then when we get the sequences back, we try and, because then we have many of them for this particular organisms, we try and stitch them together where they overlap, generate longer sequences, and we do that for every sample that we've got, and so we can see mutations through time. So when these mutations actually have an impact, they can change the protein that they're coding for, so the protein that results from them. If that happens, that's what's called a non-synonymous mutation. There's also changes in the genome that don't have an impact on proteins at all. Those are called synonymous mutations. And then we can look at the ratio between the two, and if the ratio is over than 1, then that's a classic signal for adaptive evolution. And these are the kinds of analysis we're currently doing with Fragilaropsis cylindris and with Euphausia superba, which is the Antarctic krill. Vishwadeep is the student working on the krill project, and so he's been designing an RNA probe set, or a bait set as we call it, for the ancient Antarctic krill sequences. He's selected some genes that have previously been shown to be linked to natural selection, and so we're targeting those ones specifically. So he's designed the set. He's now testing it. He's tested it on modern samples to start with to see if they actually capture what he wants to capture. They did, which is a great sign. And so now he's applying this to the extracts that we've got from around Antarctica. Prashasti, Prashasti is doing something similar. So she's designed a probe set to capture Fragilaropsis cylindris chloroplast genomes. And so she's comparing the genomes through time of this particular sea ice diatom. And so this is currently also in analysis stage, but she is generating some preliminary data. So what you can see here is that she found some of those mutations. They're mostly found in the chlorophyll biosynthesis machinery and genes. And she's started to do some selection analysis also, where she's showing that carbon fixation genes are probably the ones that are under some evolutionary pressures and where there's signs of adaptive selection. This is the last project I want to introduce to you. This is Timothy Nugroho. He's a master's student in the lab, and he's working on samples from the 2022 Collapse voyage that Laura led. He's been looking at 2 different sediment cores and has selected samples that were on a relative scale high and low carbonate samples. And so he's been doing some tests. Again, this is something that he just got some results back. They're sequences, that's great. We don't know what they are yet, but he's targeting these kinds of layers because he's really interested to look at mollusk signals in the future. And so we thought we do a first step into this direction and see what's the preservation like when we're comparing high versus low carbonate samples. Okay, this is my last slide. I thought I'll finish with something that is coming up next. I'm currently in the planning phase for a voyage on the RV Investigator. This is the Cook Ice Ecosystems and Sediments voyage, in short, COOKIES. We are leaving Hobart on the 1st of January 2026 for about 7 weeks. And you can see there's a very big team of PIs. So we're about 11 PIs, about a third of them are geneticists and working on genomics. So it is a very biologically focused research. We're going to be looking at eDNA, and we do population genetics on benthic organisms, ancient DNA analysis. But of course, none of these analyses are really, especially ancient DNA, that we need to know what's the environment, how old are the sediments that we are generating. And so we're really working towards looking at this entire ecosystem from all sorts of perspectives, collecting bathymetry data, oceanography data, sediments data, chemistry data, and so on. I think that was all I had. Thank you very much. If you have questions, I will answer them now or later.
Thank you, Linda, for this very informative presentation. We have a question there.
Thank you very much. I was wondering, the dating beyond the C14 range, where do you obtain the dates and how accurate are they?
How accurate are the 14C dates?
So these are all C14 dates? Because, I mean, you had like 1 million years.
No. This,
So 100, beyond whatever, 100,000.
Yeah. So these age models are put together by a lot of different techniques. So we do have biostratigraphy, paleomagnetism data. There's some correlations to sites, to a very closely located site, U1537, that has a really good age model for the expedition. So there's lots of different techniques that were used to date all of these different cores. Yeah.
And a second question, in this very exciting science, do you plan to continue at some point with ice sheet cores of the bed that would give us for example, collapse of the West Antarctic ice sheet and so forth?
Yes. So I do currently wait for some samples from ice cores. They're from a million-year ice core project that is undertaken in Australia. I think the communities will be very different. I don't think we'll be finding much eukaryote DNA in there at all. But I'm definitely very interested to see what we could detect. Yeah.
Okay, there is. All right. Let me first, lady first.
Thank you, I learned so much. And I'm sorry if I didn't retain this, but with regard to sampling, do you have to sample it immediately, or can you go back years later and increase the resolution of your sampling and still do your analyses?
You don't have to sample immediately, but to preserve sample integrity, it's definitely recommended. So you can take sediment cores and store them somewhere, and I've actually been working on samples that were under vacuum nitrogen gas refrigerated for 7 years, and they had very good preservation. So it's possible to keep cores. It's very difficult when you've got the long cores to do that and to keep them under such conditions. So often they, under 4 degrees, and there is microbial activity happening in those cores. So that's not recommended. There's also, if we could freeze them, but then we have water movements in the core, and that's also not ideal. So the best way to collect samples is really sample them straight away, and put the small samples into the freezer. It makes it much easier. Yeah.
Thank you very much for this great introduction into this hot research topic. I have a question, the preservation of ancient DNA. Is there a difference between terrestrial sediments and marine sediments, or do you have a better, or would the preservation in terrestrial sediments better?
Yeah, so I haven't looked at many terrestrial sediments, but the majority of the environmental ancient DNA research field actually focuses on terrestrial environments, lake sediments specifically. There's a lot of DNA preserved in those. And so I guess it's also a matter of the proximity of the organisms that are living in the water. How deep is the environment? How far is the travel time for the organisms and the dead DNA to sink to the seafloor? So just considering that, I'd say a terrestrial, like lake sediments, have probably better DNA preservation just because there's so much more. In the deep ocean sediments, we have much less. Yeah.
My question was more aiming whether salinity also damages DNA.
Right. Yeah. Okay. We don't know. So there is certain factors that we think have an influence on DNA preservation. For example, silica content, clay content. But there is so much more that that we need to find out. Yeah. There'd be good experiments to do.
Okay, somebody else. I have a question. Probably you mentioned, but I wonder what caused this increase on diatoms 14,000 years ago or something like that?
Yes, so we do think that is like environmental change that is happening around Antarctica there. And so we've done a few more analyses rather than that what I've just shown. There's really a restructuring of the food web happening from sea ice diatoms to more open ocean diatoms, so from Fragilareopsis cylindris to Chaetoceros species. So there's a whole change in the diatom community itself that seems to be responding. Yeah.
Okay. Somebody else? Okay, we thank once again Linda. Thank you very much. Just want to announce there is now available this special issue of this magazine is from INACH, a special issue on Earth science in Antarctica. Is in English completely, so take one and take it with you. Thank you. Let's go.